fruit size. Here you have the wild type hirsutum, and which has no lycopene. Here is the cultivated, and we could find some genes which are increasing the color of the cultivated one. I don't want to enter into this detail because I will not have enough time, but maybe this one, just to say that with the new technology which have been recently developed for overcrops, when using, say, 10 selected wild types, you could introduce as much new genetic diversity than when using 100 randomly identified wild plants. So, to answer the question I was asked to answer, have we top out in use of natural genetic diversity? Certainly not. There is still a lot of diversity available, and I'm sure that the new technologies in terms of molecular biology, I need to make a comment here, which is very short, which is to say, for one data point in molecular markers, meaning one marker, one plant, in certain companies, it does not cost more than 1 cent of a US dollar per data point, when it was $10 10 years ago per point. So now it makes the use of this technology easier and cheaper. So we also top out in biotechnology for conventional breeding, meaning marker-assisted selection, meaning seed quality control, or meaning cell biology. I just wish to say a few comments. One is to mention that the initiative, and you maybe know about, but I know Spain is participating to this initiative. We have called it Solanacea Genetic Network, meaning that the full genome of the tomato will be sequenced quite soon, and not only the one of tomato, but also the ones of related species, and then you could take benefit of the information you will get on a crop like potato or even coffee, which is not a Solanacea, but which is close, to get information for tomatoes. Have we top out, I mean, on biotech for conventional breeding? My answer is also certainly not. And another example is to say most of the seed companies working on tomatoes are desperately looking for what we call DH technology. DH technology is the production of a plant from a pollen grain, in order to save time for creating new lines. And that exists for wheat, that exists for corn, that exists for barley, that exists for many crops, but unfortunately still not available for tomato. 2 words about genetically modified tomato. This is a long story, and I don't want to take too much time on that. Probably most of you remember that the first commercial genetically modified plant has been launched in '96, and it was a tomato. And it was launched by 2 companies at the same time. One was Calgin, launching fresh tomatoes in USA, and the other one was Zeneca, British company, launching PG tomatoes also, but for processing tomatoes. And that has been a failure, not because of the technical aspect of the traits, because these tomatoes were effectively modified, they had the traits, and they were quite interesting. However, we were facing the public acceptance issues. So I just want to tell you a few words more about this GM issue. You have to know that today the GM seed market is about $6.9 billion US, meaning 20% of the total seed market is GM seed. No tomato, but it means if tomato comes back, maybe we will have some market for that. So again, for the question you have asked, are we top out in GM tomatoes? I would say no. We have a lot of candidate genes of interest, but the cost for launching these new GM tomatoes and the public acceptance is limiting or delaying the development of GM tomatoes. But there are some in the pipe. And I can tell you that there are many seed companies having GM tomato in the pipe, just put on the shelf, waiting for the time it will be easier to launch them. And it's not only tomato, it's even lettuce or onion. I have seen GM lettuce or onion in seed companies just waiting, yes, just waiting for a better time to launch them. So seed production, I will not tell you a lot. Simply, you have to know that we still do not have reliable cytoplasmic male sterility to produce hybrid tomato seeds. And there are a lot of seed companies looking for that, and they will probably find one day. The second thing is, you maybe do not know that for high-value tomato seed, now it's not anymore a seed which is sold to the grower. This is a grafted planted. And you have a tomato variety as a rootstock and a tomato variety grafted on it for having a better yield in greenhouses. And then there are companies which are looking for fully controlled propagation by in vitro technique of tomatoes. Why do they want to do that? Because if the lines from the parents from the hybrid have a very big value, they are more and more reluctant to put these lines in China or in India in fields where they could not control their lines for the production of hybrids. So they are looking for keeping these lines in-house and the seed production in-house by in vitro propagation. And I don't want to enter into this also because that is too much, but I can tell you that for other crops, this in vitro production is started to work very well. It doesn't work for tomato, but there are companies which are restarting R&D seed company restarting R&D project for having this seed production technology available in their house. So I have to go quite rapidly, as Joachim asked, but my feeling is the following. We surely have not topped out with the genetic improvement of processing tomato. There are a lot of technology which are in the pipe and which could be transferred to tomato. But I'm personally convinced that we will have to leverage from fresh tomatoes because there are more efforts and expenses invested for fresh tomatoes than for processing ones. And you will have probably to better coordinate with processors and end users. What does it mean? It means that this is the chain we are always looking at when looking from the gene to the consumer. And this is a coordination which has been tried here with the CTAEX. And I think it is a very positive thing to have this capability to coordinate from the early beginning of the chain down to the consumer and to feed back the seed industry or the breeders with the information coming from the consumer. I'm sorry if I have been too long.